Now we are moving into the applications of enzymes. Earlier we have seen that in almost all aspects of human civilization we have uses for enzymes. Now in this presentation we are going to see them in a little bit detail. You see, proteolytic enzymes, proteases are used in meat, seafood and other types of industries. And fat degrading enzymes, for example lipase, is used in beef and other types of food materials or during their preparation and processing. Sugar degrading enzyme amylase is used during the manufacture of chocolate, cake, biscuits and other food materials and drinks. Pharmaceuticals is a very important industry which are utilizing enzymes. For example, nitrile hydratase, transaminases, monoamine oxidase, lipase, penicillin acylase, etc. are used in pharmaceutical industries. Their applications include synthesis of intermediates for the production of active pharmaceutical ingredients. Like that. You see, in the food processing industries, trypsin, amylase, glucose isomerase, etc. are being used. These are the applications: conversion of starch into glucose, then production of high fructose corn syrup, etc. In the detergent industries, proteases, lipases, amylases, cellulases, etc. are being used. For what purpose? For the removal of stains, fats and oils, etc. from the clothes. In the biofuel industry, lipases, cellulases and silaneases are used for the production of fatty acid methyl esters and for the decomposition of lignocellulosic material for the production of bioethanol. In the paper and pulp industry, lipases, cellulases and silaneases are used for the removal of lignin, etc. You know, lignin is a material present on the secondary cell walls in plants. Often this lignin is a very tough and resistant material and it should be removed for the proper processing of those materials. Amylase is used for the hydrolysis of starch to produce glucose, etc. Cellulolytic enzymes which are responsible for the breakdown of cellulose, are used for the hydrolysis of cellulosic material to form pulp, etc. and for the sugar and alcohol industries, etc. Chitinase, an enzyme that can break down chitin, is used for the conversion of shellfish waste to N-acetyl glucosamine. You know, shellfishes are actually animals living in water. Their shells are made up of this polymer of N-acetyl glucosamine. It is called chitin. This N-acetyl glucosamine has very much important industrial applications. L-lactonolactone oxidase is an enzyme used for the conversion of galactose from whey hydrolysis to ascorbic acid. You know that ascorbic acid is vitamin C. Pectin esterases and pectin lyases are used for the degradation of pectin to produce butanol in some industries. Lactases are enzymes capable of acting on milk, and this dairy waste processing and production of value-added products involve usage of lactases, and proteases are used for the solubilization of fish and meat remains actually in their waste treatment plants. And again, amylases, invertases, glucose isomerases are there. You should thoroughly go through this list. It will come for your examination. And in your future, when you try to do some research work in enzymology, you can look for the enzymes, which are the enzymes of industrial or commercial applications. Okay, here the importance of this slide is that name of the enzyme is given and also source of the enzyme, the organism from which the enzyme is isolated, that also is given. This is a very important slide in that aspect. And these are actually different types of enzymes. You might have heard of enzyme classification. Classification of enzymes into different classes. You might have heard of it earlier. I think you have prepared slides on that one. Oxidoreductases are utilized for these types of applications. Catalase is used for sterilization of milk. Glucose oxidase is used for the removal of glucose from food materials. Lipoxidase is used for bleach in the white bread, etc. etc. Okay. And hydrolases, beta and alpha amylase is used in the brewing industry. Cellulase is used during winemaking. Glucoamylase is used in starch processing. Penicillin amidase is used in antibiotics, etc. So make a note of these slides as well. And serum enzymes. These are the enzymes present in our blood in serum, and they are used for a variety of diagnostic purposes. What is meant by diagnosis? Identification of a disease. For example, serum glutamate oxaloacetate transaminase, SGOT. Serum glutamate oxaloacetate transaminase, SGOT. Or serum glutamate pyruvate transaminase, SGPT. This enzyme is used for diagnosis of viral hepatitis, myocardial infarction, etc. So the level of SGPT is a decisive factor during the identification of myocardial infarction, or we say heart attack. Amylase is used for the diagnosis of pancreatitis. Ceruloplasmin is used for the diagnosis of hepatolenticular degeneration, etc. etc. Okay, so make a note of these enzymes and their diagnostic applications in clinical biochemistry. And these are other examples important in clinical biochemistry. Collagenase is used for the wound debridement, treatment of skin ulcers and burns, etc. You can see here some numbers are given. They are the references. Actually, I took these slides from some textbooks. So in the textbook, at the end of the chapter, some references are given. That number is given here. Already you have those textbooks with you, and you may try to understand from which book this slide is taken. I mean, this picture is taken. Again, liver enzyme, ALT, alanine aminotransferases, normally found largely in the liver, etc. Liver, heart, muscle, kidney and brain. Aspartate aminotransferases, earlier called SGOT, we found in a previous slide. Normally found in many tissues. So these are the enzymes used for diagnosis of diseases. In food processing technology, food science and technology is developing very fast. Of course, it includes applications of thousands of enzymes, not hundreds of enzymes. Chymosin, lipases, lysozymes, etc. are used for cheese manufacturing. If you go to the plant of Milma, etc. or Amul, you can see the widespread activity or application of these enzymes. Beta galactosidase, lactases, etc. are used for breaking down lactose into glucose and galactose in milk processing to avoid lactose intolerance. Some of you may not be able to drink milk because if you drink milk, you may have vomiting, stomach pain, cramping of the stomach, etc. It's because you are not able to digest lactose anymore. This problem is called lactose intolerance. It is not a severe problem. You just avoid milk, that's all, nothing else. And sometimes in milk processing industries, we break down lactose so that even people having lactose intolerance can drink such a type of milks. Okay, yes. Then lactoperoxidases are used for cold sterilization of milks. And acid proteinases for milk coagulation. Like that, you can see in different industries, different enzymes are used. And again, coming to dairy industry, brewing industry, alcohol production, baking, wine and fruit juice, meat, protein, starch. There are variety of industries are there which are using enzymes. You find out their applications as well. And again, nowadays some types of plastics are being used. What type of plastic? They can be actually uh, used for degradation. I mean, those plastics can be degraded. This when the plastic is made along with the, I mean, using polymers, enzymes are also added such that once it is thrown away, it will be self destroyable. That means it will decompose by itself, so it will not be a harmful additive upon the environment. You see how the science and technology is advancing. Zero waste. Enzyme incorporation into the plastic material is resulting in zero waste. Bioremediation. What is bioremediation? Removal of contaminants from the environment using living organisms. Friendly bacteria in spill-away products eat oil and hydrocarbon waste. These microbes digest and metabolize this waste, turning it into water and harmless gases. Finally, the microbes release water and gases back to the nature. You know what happens? Microbes are engineered. Microbes are genetically engineered to incorporate specific genes of enzymes so that they can eat away the harmful contaminants of the environment. And these are some enzymes having important industrial activity, industrial uses. So make a note of all those things. Refer textbooks to get more examples and applications.